Okay, so now that we've gotten that concept down about what we need to do with our food, which we'll review one more time, once we eat our food and we swallow it, the first things we need to do is break it down so we can liquefy it into something that can go into the plasma of our blood, head on out to our cells, and be used for all sorts of things like cell respiration and growth and repair of themselves. So therefore, in order to break it down, we're going to need a few organs for that. And then following that, we're going to use a few organs to kind of absorb it into our bloodstream, whatever we could liquefy, including water. And then finally, what we're going to do is have a little bit left over that was not absorbed into our body, and that is what we're going to eliminate from our body as poop. And so what I want to do first today is have a demonstration. I have a little guest for you. It's a long lost colleague of mine, very, very prestigious doctor, and her name is Dr. Seeless. And so I'm going to turn it over to her and let her explain to you one more concept. And before I set you off on that one, it's this. How broken down does our food have to actually get? Is chewing enough? Is it grinding it up enough? If we get it down to the single molecule, let's say of starch, is that enough? Let's find out, because that's what Dr. Seeless is going to talk to us about. Hey y'all, I didn't know y'all stopped by. Mrs. Seymour said you'd be telling me that you needed to know a little bit more information about the digestive system. So here I am, I'm going to show you a few things. Today we're going to do a little bit of a starch test. Starch, do you know what it is? Because if you don't remember, starches are long chains of sugars. You know one sugar, that's called glucose. Glucose is the one that the cell respiration is looking for in the mitochondria of our cells. So, if we could turn starch into sugar, then I know that my mitochondria are going to be able to do their job. So, let's see. When we talked to Miss Seymour, she was saying how small does small need to be. So let's take a peek at that, all right? I'm going to take a little bit of cornstarch. This cornstarch, well, it's just a powder. So, let's get a cup. Put a little water in it. Let's add a little bit of starch. Cool. Mix it up. So now we got some starchy water here. So let me show you something about starch. Starch has the ability to show itself in another way. And that way is with iodine. You guys remember, you learned about iodine with the cell because you used it to dye your cells so you could see things like the nucleus better. But here, we're going to use it like an indicator solution. An indicator solution shows the presence of something. You used that once before. You used it with Benedict's solution. Nope, you used it with bromothymol blue when you guys exhaled your bubbles into that beaker and turned that blue water green. Well, iodine takes its kind of orangey color and it turns it a whole different color when it comes into starch. So let me show you that. Here's our brown color starch. We're going to put it down here. And there it goes, already turning colors. And if I give it a little bit of help, voila, sounds like magic almost. But it is not magic. What we are looking for here, now that my accent has changed, because you know, I'm from all over the world. Now we have our black kind of liquid just because we added starch and we also added iodine. So let's take that little step further. All right, what I have here, I have 2 test tubes. These test tubes both have funnels in them. Okay, so what I'm going to do is I'm going to put a little bit of starch, no, said that wrong, a little bit of iodine in the bottom of each test tube. All right. I'm also going to add a little bit of water in each one. All right. Now, got a question for you. If I pour some of this black starchy water into this funnel, you think it's going to turn this black, or you think it's going to stay the same color? Well, let's find out. Well, looky what we got here. Same color again. Starch touched the iodine, and it's that nice dark color. All right. Well, let's change it up for the second one. If starch is going to get to our cells, it's going to have to be able to be a liquid. I got it in the liquid right here. But what if it doesn't dissolve in that liquid? What if it's just still kind of in the liquid, but it's not really liquefied? Could that be a problem? Yeah, it can. Because if we can't get it to our cells in the liquid of our plasma, it's going to have a hard time getting there. So, let's do a little modeling. Let us take, at this moment, this thing called a filter paper. This filter paper is kind of like a coffee filter, where it lets liquids through, but any solid particles will be caught within the filter. This would represent the cell membrane. Liquids pass through just beautifully. However, anything that is not a tiny particle dissolved in the liquids will not be able to get through the cell and into the cell membrane. So let's take a peek at what would happen if I poured this liquid through this membrane. Alright, I'm going to use the funnel to do that. The first thing I'm going to do is fold it into quarters. You can see it's almost looking like a funnel already. Then from there, I'm going to take it apart and turn it into a little cone. However, it doesn't want to sit in there. I have a remedy for that. Just a little bit of water will help this little fella sit where I want him to sit, just so that he stays still. There we go. All right, so let's do an experiment. What do you think might happen? If I put this starchy water into this funnel with the filter paper, if it's liquefied, it should pass through and look like that. But if it's not liquefied, it won't go through, and I might find all that black stuff up in the filter paper. So let's see what happens. Alright, so let's let that settle for a moment. We'll just pretend that time has gone by. You can see it dripping right here. So far, I don't see any darkness. Let's give it a little more time. Well, I think we're starting to get some good results here. How about if I mix up the tube a little bit? Let's actually stir it a little bit. Make sure we don't have the reaction. Well, I think we've got the idea here. Looking at that,
I don't see any starch in the tube. Where is the starch? Do you think the starch is in here? Well, let's take a look. Just don't want to spill it all. All right, take a look right there. All the starch, still with the iodine attached to it, is right here in the funnel, which means it's made of little tiny particles. Particles so little we can't even see them. However, these molecules of starch are just far too large to pass through a cell membrane. So therefore, how small can small get? Even smaller than a big molecule like starch. These big molecules like starch, we have to break into little molecules like, you got it, sugar. Sugar does dissolve in water, doesn't it? Well, that will be our next test that we will show you next time. So I'm so glad I got to meet you guys, and so I hope you can use all this when y'all talk to Mrs. Seymour. All right? See you later now, guys. Bye-bye.
Oh, thank you so much, Dr. Seeless. That's exactly the point I was trying to say. Breaking down food. How far do you have to break it down? Farther than you might think. Even a molecule might be too large to pass through a cell membrane. And so large molecules like starches, which are some of the biggest ones we eat, need to be broken down into smaller little subunits. And what were those subunits called? I'm hoping you're saying sugar. Sugar is exactly what we're going to talk about here. All right. And so tomorrow we're going to have a quick little lesson and a quick little demonstration that is going to turn starch into sugar. And so if you guys happen to have any saltine crackers, maybe oyster crackers, kind of like a doughy kind of starchy food that doesn't have much flavor to it. If you can bring that to tomorrow's class, and if not, no big deal, because I have plenty of props to get the idea across anyway. But that is where we're going to go next. We're going to take a moment tomorrow to see the idea of how we can turn a starch into a sugar before it gets absorbed into our bloodstream. All right, so don't forget the Google form. See you tomorrow, and have a great night and stay healthy. Bye-bye.